Station. Hey, hello everybody. Thanks for coming today. I think this is the last department seminar for the semester, so thanks very much for coming. I'm pleased today to introduce Dr. J Gidmark. He's from Knox. I'll say more. He gave me the best biography information I've ever received. I cannot do it all. I will tell you that Dr. Gidmark grew up in Minnesota. He has loved biology since he could walk. He's been catching frogs and eating slugs since the time he was 3-ish. I think that's even before me, so that's awesome. He was an undergrad at the University of Minnesota. I got a double major in fisheries and conservation biology, where he studied phylogenetics or systematics. He got his PhD from Brown University, where he studied functional morphology and biomechanics of carpharyngeal jaws. Does that mean here?
Thereabouts. Yeah.
Okay. So he got his PhD at Brown, then did 3 different postdocs, which is a sort of like additional training you often do before you get a professor job. One was at Brown University, one was at Friday Harbor Laboratories at the University of Washington, where he was doing research on salmon jaws. A third postdoc at University of Chicago, where he studied jaw and tongue motor control in macaque monkeys. He currently works at Knox College. He's been there 10 years. He's an associate professor there. The classes he teaches are intro organismal biology. He teaches upper division classes like biology of fishes and comparative vertebrates, which is anatomy. What else do I want to say? He has over 24 peer-reviewed papers. He has dissected and eaten a lot of creatures. His least favorite surgical subject is a turkey. His most delicious research subject was the king salmon. He's recently shifted his lab's focus, so you heard me, he's done a lot of fish functional morphology. He recently shifted his research focus to the utility of model organisms in clinical research, so human research. And his title is On the Relevance of Biomechanical and Kinematic Variation in Mammals for Clinical Jaw Joint Studies. Thanks.
Okay. Thanks for having me. Thanks for coming to hang out and spending a little bit of time here. And thanks for the invite. I really appreciate it. I'm honored to come. I want to start first with actually my some noises. And my acknowledgement slides. These are pictures of some of the people that are in my lab who've been working with me at Knox, which is an undergraduate-only institution. This is a smattering of them who've been with me for the last couple of years. And I try to do what I can to be an empathetic mentor and to try to help foster new science, but make no mistake about it. These people are the engine of the entire lab. All of the research you're going to see today is because of them and thanks to them. And they always teach me more than I ever teach them. So we're talking about feeding today, in case you didn't guess on that. And my understanding is that we will get to feed together after this, right, Sean? There's a, I think so. We got some food? Okay. Great.
1 o'clock and 365.
There you go. Okay. Oh yeah, there it is. That's us. Yeah. So just to start, I want to show a video of one of my best friends in the whole world. He's my roommate in grad school, and he's eating a burrito. And as he's eating, I want you to think about all of the different tissues that are involved, all the different pieces that are involved in his ability to procure and deal with that food. You saw his tongue there for a second, his teeth, which are actually 2 different kinds of hard tissues that are different than bone. He's got a jawbone, he's got a skull bone. There's ligaments that are holding this whole system together. There's muscles that are moving those pieces all around. And all of those have to be really working perfectly in order to have this thing work. So I'm going to write a few take-home messages up here. And the first one that I want to have is that feeding involves, can't spell today, complex, interlocking parts. Right? All of those have to be in the right place, doing their job in order for feeding to work. If his tooth was down here, the burrito would win, not him, right? You really have to have all these pieces together. And so in that thought, we're gonna try to expand out, not only thinking about how we have a bunch of different individual parts, but there's also a lot of diversity out there. There's a lot of different animals out there, and we're gonna focus mostly on mammals today. So can we get a little audience participation? Somebody give me a mammal.
Beaver.
A beaver, that's a great one. A couple more. Cows, those are good. So think about beaver versus cow. Beavers have these big front teeth. Cows actually don't have very much. And they do have incisors, but most of their jaw is back there. Give me a couple more.
Aardvark.
Aardvark, yes. I love that one, right? Super long, skinny head. One more. Wolves, yep. So again, different kinds of foods, right? A wolf is eating meat, so it's going to have sharp teeth. The cow is going to eat flat things, and that's the diversity there is pretty impressive. And I was sitting down with my student Alyssa to write a she was giving a presentation a few months ago, and I was sitting down with her to help her write the talk. And she's an honor student, so she came and had all this like stuff all ready to go. And this was the title of her talk, On the Clinical Relevance of Animal Variation. And I looked at that and I was like, that's pretty bold, right? Like that is like, how do we talk about the biggest possible scope that we could have? But then as we started to go through the talk, I thought about it and I was like, you know, Quinn, that's not bad, right? That's like really what we think about, what we need to be thinking about is this huge variation that there is across all of these different animals. So the second kind of take home I want to leave you with is not only does feeding involve complex interlocking parts, but different species vary in their anatomy. And I can't spell anatomy. Spelling never counts in my classes. Okay, and it's really true. There's this huge diversity of out there across mammals. There's differences in ecology, there's differences in diet, there's differences in evolutionary history. There's all these amazing differences that I would argue result in this huge variation in anatomy. Not only that, but there's also variation in how that anatomy moves. Here's just a couple of ones. A couple of gifts for us to look at. Here's a cow that's chewing side to side. It's got a very cyclical motion of one like left to right, right to left. That's a kangaroo over there, actually totally different, opposite. Instead of moving right to left, it's going front and back. So not only do you have complex interlocking parts, not only do you also have huge variation in anatomy, but you also have huge variation in motion. So if you have some set of parts that are working together well, but they're moving wrong, then you also don't have anything to work, right? You really got to, again, be thinking about on the variation of all of these things as they work together. It turns out studying motion is nothing new. I didn't invent it. These are some still frames from Muybridge's work in 1887, so many, many years ago. And he published a set called Animal Locomotion, just like how animals move. And I want to point this out. It was an 11-volume set. Lots of data, right? You could explore animal locomotion, animal motion for your whole life, and you'd never scratch the surface of it. So we got to think about like, how do we do that like most effectively to get some cool stuff done? So one of the ways that we often study animal locomotion or animal motion, I would say, is this really interesting technique called XROM. It stands for X-ray reconstruction of moving morphology. It's a technique that was in development when I was a graduate student at Brown. I was part of the development team on it. And the short version of what it is you have a couple of X-ray video cameras, maybe a couple of other sort of light cameras, and you're watching an animal do something. In this case, from this Cambeck et al. Paper, it's watching a partridge walk around. They were interested in the hips of these birds and how they work. But in theory, you can put any animal in there and you can track the way it moves and try to understand something about its motion. The heart of XROM is based on taking a surgical approach to implanting little metal spheres, little metal, I mean, they're not beach balls, but they're tight, into an animal. So these red dots are highlighting individual markers that we've surgically implanted, in this case, into a pig. We then take a CT scan of that animal so that we know exactly what shape that animal is and exactly where those markers are in that shape. And then we use those two individual data streams, put them together in a digital environment, a digital animation environment, to actually reconstruct 3D motion of these bones. We've done tons of really cool stuff with this. This is a video from one of the first XROM studies, which was of a pig. You've got X-ray video cameras here and here. We're going to put the pig in there, and then you can see those are the videos that are being generated from this X-ray camera so we can watch this pig munch on its food. So we've learned tons of really cool stuff from XROM over the last 15 years. I'm just going to run through a little smattering of them here. This is a situation where this is that same thing that you were just looking at. We can now zoom in to individual parts of that animal. So here's the skull from a ventral view, from an underside up view. And if we zoom in just on this little inset here, we can look at, here's this tooth, left and right tooth. And we can track the motion of the other jaw. So these little red and what is that, green cyanide? I don't know colors. We're going to track the motion of those tips of the teeth as they drag across the teeth on the upper jaw. What we showed from this is that this animal is actually occluding both sides of its jaws at once. It's chewing with two sides of its mouth. If you think about like eating like a pig, it's because they're chewing twice for every one of your chews. Pretty cool take home from that paper. Here's another paper that I worked on. I'll say that any of these citations that are underlined are ones that my students and I have worked on or that I've collaborated with. When I was doing my work with monkeys for my postdoc, we studied a bunch about the tongue and the muscles involved in that tongue in monkeys. And we studied also there's a bone right here called the hyoid bone. You've got one too, like right in the base of your tongue. And we were looking at the mechanics of how the tongue produces swallows and how it helps with chewing. We showed that it was a good model system using that X-ray video approach. Another cool one that we did was we put a bunch of markers into the tongue. Again, this animal like variously, so they don't know that we're like shoving markers into it. But we looked at the way that that tongue moves over time during chewing and swallowing. And it turns out your tongue actually curves, like it curves over and then pushes to the side to move food around in order to keep the stuff that you're chewing on actually stuck in your teeth, stuck between your teeth, I should say. Here's another really cool one that I like. It's a pretty recent one from a graduate student at U Chicago who I was a collaborator with. And one of the things he showed was that depending on the posture of how you're standing, the tongue base, the hyoid, that bone on the base of your tongue, is going to change its position. And that's super important that you're going to need to know, or you're going to need to work right as we're eating some, what is it, pizza, yeah? Pizza, yeah, good. Maybe we should text them to make sure we have it. But I want you to think about it when you're doing that. And so as you're thinking about maybe we're going to eat some pizza, just get a little saliva in your mouth and swallow. And it's like kind of not that hard, right? It's like normal. But if you do it again, and then you put your foot down, and you try to swallow, it's hard. It doesn't work right. So it turns out, because of this set of complex interlocking parts, and because we're going to have a lot of motion, we have to be in the right posture in order for swallowing even to work at all. If you tried to swallow all of your food with your foot down like that, it's not going to work. The burrito or the pizza is going to win, right? So what this picture or what this paper showed was that depending on whether you have a extended head or a flexed head, that hyoid is going to move in different places relative to your skull. So we have to be in the right posture. Take home from that. This is a great paper that my student Emily McParland headed up, and I think she's on Zoom. Hi, Emily. And she, one of the things she showed was that during occlusion, when teeth are squished against one another, when they're doing their job, in rats, we were able to quantify how much motion happens and what direction it happens in. And so we quantified for the first time, she quantified for the first time, that how much forward, backward motion happens. And that's the primary motion during the business end of a rat chewing, which is totally different from, say, a monkey or something else. So now, what do we do with all these data? So like, we have this cool technique. I've told you, like, I just went and explored for a bunch of time, and my collaborators and I just kind of like did a bunch of random stuff. But we want to like maybe make something useful out of our lives. That would be great. Let's like get some overall greater punchlines. And so if the question is who cares, the answer should be you. Does anybody have, or is anybody willing to share that they have TMJ? It's a problem with the jaw joint. Turns out every single one of you has TMJ. The TMJ is just the jaw joint. It's the temporomandibular joint. It's the joint between your mandible and your temporal bone. People talk about this often because of temporomandibular diseases, or TMD, where you have like jaw clicking or something like that that happens in your in your head. We'll talk about that in a little bit. The temporomandibular joint itself is made up of a pretty complex web of different soft and hard tissues. So you have the condyle. I got my skeleton here. In the jaw, you have the condyle right here, which is the ball of the ball and socket joint. You have the temporal. Oh, head's gonna come out. Careful. You have the temporal bone, which has a socket right there, and so those together form the ball and socket of the TMJ, the temporomandibular joint. And you also have this series of different ligaments. You've got this soft, cushy, articular disc pillow that is helping to absorb shock. You also, of course, again, have all these muscles that are there doing their job as well. And importantly, you need to actually be able to move in a whole bunch of different ways in order to have your jaw work. It's not just this simple hinge that goes, right? As we've already shown, there's a little bit of back and forth. If you're chewing on a piece of gum, for example, you're not just going up and down. You're doing some side to side. You're grinding on that material. And when you have a problem with your TMJ, that becomes what we call TMD, or temporomandibular disorders. These are a whole host of different things, from, like I said, jaw clicking. There can be oral facial pain, trouble swallowing, trouble speaking, all kinds of different limitations to range of motion, and lots of overall problems. One issue that comes up a lot is that there can be a restriction in the range of motion. You can't open your mouth as wide as you otherwise could. So here's some traces of motion of a couple of different subjects that are opening and closing their jaws. And the green lines here are denoting the bounds of a control group, how much motion, how much rotation is going on in normal healthy species. And then the pink here is a restricted jaw opening amount of rotation that's going to happen in a pathological or someone who's got a TMD problem. So we have a restriction of range of motion. We can get cool data like this and some other kinds of data through some very complex sets of apparatuses. This image on the right is from a group in Switzerland who has developed this really complex engineering way of calculating motion of the jaw. But you can imagine, like, if you're wearing this whole thing, like, are you chewing really normally? Like, maybe. We can't verify, right? It's kind of challenging. So the only other way that we have studied chewing in humans is through modeling, and that's the main other way, I should say. And so these are great approaches, but there's only so far that can go, right? We can't always get all of the information we want from those kinds of data. So most biomedical research has then transitioned to using what we call model organisms that we can do experiments on. We can, in something like a rat, for example, we can induce temporomandibular joint disorders. We can breed rats that don't actually have like an articular disc, for example, and we can study those kinds of differences. Lots of these different species are useful in different ways. Some smaller ones are sort of more tractable experimentally. The bigger ones, like maybe a monkey, is really anatomically close to us. We know a lot about the neuroscience of sheep, for example, and so this set of 6 organisms is a really common group, set of organisms for us to kind of play around with. And so what I'm going to talk to you about today is 3 major studies that we've conducted. I'd love to talk about 14 of them, but we don't have time. Somebody wants me to keep the 40 minutes. So we're going to stick with these 3 major questions and 3 specific students who kind of headed them up. And we're going to talk about whether we can use this motion technique to improve precision in the ways that we are measuring motion. We're going to try to understand how these common model organisms are moving differently. And we're going to try to sort of scratch the surface of temporomandibular joint function and talk about how it varies across species. So the first research question was headed up by Emily McFarland, rock star for the record. She's part of 3 different papers out of my lab, and also did 2 papers as a postbac for 1 year in the University of North Carolina. Just saying, she's in her 2nd year of her PhD at Brown. And her honors thesis was trying to tackle this idea of, is this tool really actually going to improve our ability to resolve motions in TMD-related things? So what she did was that she took the heads off of some animals the Nescus worthy is fine, don't worry about it. And then she surgically implanted these markers, froze them, vacuum sealed them, and then did the whole XROM process on that head. I know what you're thinking. You're like, Nick, if if the head is cut off the animal, and if it's frozen, the jaw is not moving relative to the skull, right? You're 100% right. We know it's not moving, a priori. The question here, though, is this technique going to allow us, how precise is this technique going to allow us to understand position? So what she did then was she animated this using XROM, and this is the skull of a macaque that she used this technique on, and you're seeing right now two jaws superimposed on one another. One is the standard position from the CT scan, and the other is the reconstructed position of that jaw over time. So what you're seeing here is noise. This is the noise inherent in the XROM approach. And what we're trying to say then is this any better than other tools for measuring motion? It turns out it's a lot better, like way better. So here's a series of different papers. The citations are here at the bottom. The filled rectangles are how much motion happens in a healthy individual. And the open squares are what happens in a pathological individual. And these are three different sort of cardinal directions, moving forward and backward there on your left, moving up and down in the middle, and moving side to side on the right. Now, the error bars that are in pink are the error bars that were from that study, the error bars that they found, their precision. And I want you to note that in the upper right, the key there, that pink is the standard deviation of their error. That's how finally they were able to resolve those differences in motion. And I want to point out that if here's your error bar for healthy, and here's your error bar for pathological, those two aren't different. We're actually not able to resolve differences in healthy versus pathological. And yet Henderson et al. Published that. Somebody needs to talk about stats. And indeed, in all of these different individual motions, we're not able to resolve healthy versus pathological species or specimen. Our data is not calculated by standard deviation. These error bars are range, which is going to be way, way larger than standard deviation. Standard deviation is going to be much tinier. Check out that error bar here versus here. So what we've shown here is we are indeed not only able to match resolution in other motion capture techniques, but we're actually going to be way, way better than it. We're able to reconstruct motion at a much finer scale to the point that we could actually resolve healthy versus pathological individuals, at least in terms of front-back translation, and probably in this side-to-side translation. Wicked cool stuff. So the next thing we did was, it turns out that this monkey data I published with Courtney Orsman and some of our colleagues a couple of times in 2018 and 2020. Rachel Menegez's original study on pig XROM was published in 2015. And then we have McFarland et al.'s study out of our lab in 2018. We actually have 3D motion of 3 out of those 6 species. Rabbits are coming too, we'll have those too. But for the moment, we have these 3 pieces of information, and we're gonna rigorously now quantify in an objective way that sort of equally treats each of these groups how much motion happens. To do that, we're gonna fit a plane to the occlusal surface, basically fit a plane to all the tooth cusps. We're gonna use that to orient what is front back, or what my anatomists might call anterior posterior, superior inferior, that is up and down, and medial lateral, which is left and right. And so as we're trying to quantify that, we can put, we can actually measure that around the condyle, around the ball of that ball and socket. And we can individually then decompose how much motion happens in each of those cases. So we can start with translations, right? That's just straight inline motion. So here would be anterior, posterior translation. Here would be superior, inferior translation, up, down. And then if I turn it towards you, you can get medial lateral translation. I haven't rotated this jaw at all. It's just sliding in 3 different orientations. The beauty of XROM lets us also measure rotation about each of those axes. So if I'm measuring rotation about this front back axis, we call that roll, and that's where this jaw will go like this. It'll rotate about an axis that's running anteriorly. We can do yaw, which is rotation about that superior-inferior axis. I'm doing that. And we can also measure pitch, which is rotation about that medial-lateral axis. So doing the normal, what we think of as opening and closing the jaw. And we can now decompose all 6 of those motions one at a time. So let's do that. We also can not only measure that movement of that joint in those 6 degree of freedoms, but just like that pig study I showed earlier, we can measure translation of a landmark, some anatomically relevant thing like a tooth cusp relative to the skull. And here, if we just look at our rat data from 2024, we can see that we have a little bit of rotation around those other 2 axes, but most of our rotation is that pitch, is that moving up and down. We're going to zoom in on just the times when the jaw is elevated and those teeth are in occlusion. And we're going to track that by zooming in here really closely into this little jaw space. And we're only going to look at that yellow region up there when the teeth are doing their thing. This is a research question that was asked by Nan Kim, who's going to dental school next year. And she was interested in what is the variation in the range of motion of these 3 different model organisms during the occlusive phase. So when the jaw is, when the teeth are occluding each other. What she found was that we actually have a huge amount of translation in rats, both front back and a little side to side, way more than we have in either of the other 2. This y-axis is scaled as a fraction of the head size. And we see that in both pigs and in monkeys, we have predominantly side-to-side motion, which kind of makes sense, like as you're chewing, kind of going side to side. In a rat, not only do we have more side-to-side motion, but we have even more front-to-back motion. So it's a fundamentally different pattern of motion in this group. Here's a translation of the condyle itself. So that's that ball in the ball and socket. Turns out that ball moves a lot in mice. It actually turns out that because of the anatomy, there's not a lot of constraint in that socket. It's a regular ball, but it's a big socket. So there's a lot more slop in the way that lower jaw moves relative to the skull. Just so that we're like kind of having a reality check, we also measured rotations during that time. Again, X, Y, and Z. And one of the things you'll notice is that we're like plus or minus 2 degrees. So we're not having a lot of rotation happening during the occlusive phase. Basically, we're sliding the jaw forward or sliding it side to side, not a lot else. Okay, so Nan's take-home points then were that in general, we have a lot of anterior, posterior, front to back translation in a rat, whereas we have predominantly side to side translation in pigs and monkeys. And because of that, rats might not be a perfect model organism for studying jaw joint disorders. Maybe in some specific cases, but at least you gotta realize that there's a limitation to how similar to you a rat is. Shocker. Okay, so then in addition to not only these hard tissues and how they move, and the technique overall, we have a bunch of soft tissues here, right? So here's a rabbit. There's a few different pieces of this muscle that we call the masseter. That's your cheek muscle. If you clench your teeth and touch your side over your cheek, that's what we'll use a lot when we're eating our pizza today. There's also a couple of different pieces of the temporalis, which is the one if you push on your temples and clench, there's like a little bit of bulging. There's also a couple of muscles called pterygoids in there. We can get into all that anatomy, but long story short, there's a lot of soft tissues that are there. One of those soft tissues is muscle. It's kind of what we think about typically when we're looking at these things. But there's also all kinds of other soft tissues. So here's a zoom in, still of a rabbit. We're going to kind of slice right here and zoom in right there. So not only do you see the condyle, that CL, that's the ball, again, of that ball and socket. Here's your glenoid fossa, the socket of that ball and socket. You have this big articular disc, that squishy little fibrocartilaginous cushion pillow that's in there. And you've got a bunch of other labels in connective tissues, tendons and ligaments that are in the system too. We take that disc out and look at it. We have a kind of a cushion shape, and then we have no less than 6 different ligaments that are attaching to the skull and the jaw and the disc and sort of keeping everything in order. This is not an anatomy class. We're not going to have a test on which one goes where. Just know that there's a bunch of them. That's all I need to know. And those are really important because if that disc is not held in place, if it slips out of place, that's how you get things like lockjaw. We call that disc derangement, and that's a big deal. That's going to cause osteoarthritis, that's going to cause pain, it's going to cause limited range of motion. Sometimes that's why people get their jaw locked closed or locked open. It can be a really big issue. So to study it, that group in Switzerland that I was talking about, who had that weird, like crazy contraption on the face, they've developed a metric called the stress field, where they're tracking where the closest squish is for that pillow and how it moves across the chew stroke. So what you're seeing here is a top-down view. Here's your ball and socket, your condyle, and you're looking through the fossa. That's this sort of lighter color. What this point is the closest point between the skull and the jaw, and the condyle, I should say. And this is kind of the area around it that is most squished. That's going to move over time. So this is early in the chew stroke. This is later. Don't worry too much about the orientation. Just take it. Just the thing I want you to take home is that that's going to move. Exactly where that disc is most squished is going to move over time throughout the chew stroke. Here's the problem. That has to use this complex, crazy piece of hardware on the jaw. And that's not very transferable to a rabbit, right? You tell a rabbit you want it to wear this thing, it's gonna like not eat, right? We need a different way to try to measure this kind of a thing. And so what Alyssa did for her honors thesis is she actually asked two separate questions. She said, can we use XROM to make a metric like the stress field that is more objectively comparable across species? Shocker, we can, or spoiler alert, I should say. And then if we do, how does that actually function across different species? So she, along with a fantastic colleague, Armina Manavzada, did a bunch of code writing in this software program that we use to actually reconstruct where the closest point is of these two bony surfaces. And we can do a couple of different things. We developed this metric. I say we, I didn't write it. They did, for the record. And we can measure a couple of different things. One is that we can measure how much does that center of the stress field translate over time. How much does it move? When we do that, we don't see a lot of differences across the chewing stroke of these individuals. They're all moving sort of the same amount. They do move, right? They're not at zero, but they're all moving kind of the same amount. The other thing that we can do is we can ask where in space, i.e. where in that socket is that closest point. So if I have some colors here. So if I have my socket here, the glenoid fossa, have my condyle here of the jaw, that would go over this way and munch. Is that closest point over here in one end? Is it superior, inferior, or is it on the front side? Where is that nearest point in the socket? It turns out that is hugely different across species. So in a pig, that is all the way up front. The pressure point is toward the anterior of the ball. In a monkey, it's actually in the back of the fossa, and in a rat, it's somewhere kind of in the middle. It turns out the reason for that is anatomy. So this is an underside image of a monkey skull. Here is where the socket is, and there's this really big posterior eminence, it's called, a really big process that's hanging out right like this in the back of the jaw joint, and that's going to pull that closest point to here. When we look at a pig, which is totally different, they have like nothing behind here. That jaw could slip out no problem, but there's a really big anterior eminence. So in a pig, that anatomy is saying right here. It actually makes that noise. I don't know if you knew that. And so it's pulling that closest point toward the front. Fundamentally different impact on the mechanics of that jaw joint. And so Alyssa's overall conclusion was that not only have we developed, now again, not me, they, but I get to play along, I get to be jazz hands. We've developed this tool for actually robustly, comparatively, objectively looking at this metric across different species. And we've shown that there is a huge importance of motion. Motion is going to matter, but really it's the morphology that's fundamentally changing the way that this works. So to add to our take-home messages here, we have complex interlocking parts, different species vary, huge variation in motion, and we know that that motion is important. But we also know that morphology or anatomical shape is most important. So in the interest of, and this is something that we've looked at over and over and over again in a bunch of different systems. So this is Saxon Alvarez, who graduated last spring. She was looking at this overall spaces in that joint. This is Amira Siddiq. She's graduating this spring. She was looking at muscle orientation and torque. I just talked about Alyssa's work. This is Rosalie Ross. She also graduated last spring, and she was looking at ligaments and how they're connected. I'm not going to explain overall how these, or in the nitty-gritties of how these plots work, but basically the colors are different species, and all of the dots are within one individual. So we have motion represented by all the different spacing of those orange dots, and how close they are together means how similar they are. And then there's motion there that's important, but then the difference across species is much bigger, matters more than the amount of motion that that animal does. Same thing here. All the rats are here, all the pigs are here, all the monkeys are here. There is variation between individuals and across the chewing stroke as we move, but there's much bigger differences between species. Same thing here. Here's all the rats, here's all the pigs, here's all the monkeys. Differences happen in motion, but there's bigger differences in the anatomy. So to the extent that we ever argue in my lab, this is what that argument looks like. Somebody says motion matters, the other person says, meh, morphology matters more. That's pretty much it's the alliterative version of the way that our arguments work in our lab. So the major take-home points then, I've got them all up here already, but we have this complex system that involves a lot of different moving parts that have to work together. Those different parts look different in different organisms. And depending on how we employ Exron, we can actually use it as a great tool to explore those differences. What we find then is that while morphology is hugely important, and as an anatomist, I will always keep morphology in the utmost of my heart, but really, or I will always keep motion in the utmost of my heart, but I get the pleasure to say that morphology is really the most important thing. So as you're looking to go home today and talk to your cat, think about those differences in shape. And the differences in shape are going to be fundamentally important to the way that we're able to move and the way that we're able to do all the things that we do day to day. Got to end with a shout out to the lab, who are just lovely people. And thanks to you all for chatting. Nailed that 40 minutes, I'm just saying.
Okay, that's great. So plenty of time for questions for Dr. Gidmark. Yeah.
Hi.
Hi.
I'm an ornithologist, but I'm currently teaching a zoology class. I have a couple of my zoologists here.
Okay.
Thanks for that. I wanted to ask you a question, if you could, about the adaptive significance of these differences between species. Why do we have the posterior and pigs have
Posterior versus anterior eminence. I think if I could answer that question succinctly, that would be a Nobel Prize, which I don't yet have, and I don't find it likely. So I think that a lot of it, of course, a lot of it has to do with diet. And I think I have, let me see, where's my, too many extra slides. I'm gonna have to, I think there was a, no, maybe, no, I don't have it. So there, in a rat, for example, there's essentially no constraint. It's like they can't move. There's a side. There's guardrails on the sides, and there's stuff in the middle. But front to back, there's just like it's an open playing field. And I think that's really important for those anterior posterior motions. And that's, I would argue, probably the result of what it eats. It needs to grind up seeds and stuff. So you need a lot of throw. You need a lot of motion. And the way that other herbivores like cow, for example, deals with it is it's got a really long jaw. And so, actually, I do have a slide that might tell us about, oh, yeah, no, I can't see it in there anymore. Ugh, I'm all over the place. So in a, oh, there it is. In a longer jaw, relatively small motions of the condyles result in more movement of the teeth. So the way that a horse or a deer or a cow deals with that is they just have a long jaw. So that same amount of side to side movement is really useful for translating the teeth, which is why probably most herbivores have that really long head.
So this is just like the day. So over at Knox, you have colonies of rats and pigs and monkeys.
Yeah.
Okay. So
No, that's the beauty.
yeah. Where are you getting yours?
Sure. So there's two pieces to that. One is that the beauty of XROM is that once you have collected the CT scan and the videos, you can continue to explore that data set for years on end. So the pig data set, that was collected in like 2010. And it just existed. And it was used at that time to study the mechanics of how pigs are chewing. And then here comes Nick 15 years later to be like, we can re-examine that and look at the condyle, look at the shape of that interaction of the jaw joint to the skull. I didn't have to collect any of that data. I had to do a lot of analysis for 4 years, Emily will probably tell you. But I can use that data again and again to ask different questions, to look through different lenses of, let me attach muscles, let me attach ligaments, let me look at these different pieces. So that's like one column, is just like the fact that data can be reused.
And is that your collaborators, or is it publicly available?
So most of the journals that we are publishing, that XROM data gets published in, have a stipulation that the data have to be publicly available, like on Dryad or something. And so you have to know how to use it. Like the act of doing XROM is a non-trivial entry point, but all those videos are there, all the CT scans are publicly available. The other side is that I have this incredible collaborator named Dr. Callum Ross at the University of Chicago, who I did my postdoc with, and he's got an X-ray facility. So like Emily's precision data, she collected all of that because she could go there and collect it there. And that's part of why her honors study of that precision was all dead stuff, right? So we didn't have to have animal care, we didn't have to have any of that stuff, we just had to procure a bunch of dead rats. I say just, Emily spent like a whole year procuring all the animals, but yeah, good question. Yeah.
So I've been thinking during much of your talk about what could be used for markers in people. I mean, I guess I can see why nobody would want to have little BBs installed. But people have all these dental appliances and stuff. They're huge compared to those, maybe. But if it's something that stands out against bone, is that something that could be used?
Totally.
Or is there too much X radiation?
So you're asking a huge question unknowingly. And actually the markers that we use to surgically implant into animals were originally developed, like the metal tantalum is an alloy that was originally developed as a marker for osteoarthritis or for orthopedic research. So they were implanted into people, into like, oh, I did surgery on the knee and I'm gonna put it right there, before we ever used it in an XROM context. So that's actually where we got the inspiration from. So there are people walking around out there with tantalum markers in their bodies. Getting a permission to actually do that surgery just for this science, as opposed to in a clinical context, is harder. But that's been done. You're 100% right that dental implants and things like that will work. There's also, one of the things we played with a little bit was the idea that if we could use like a wax or something that we could implant them with and like put it on the sides of the teeth. For now,
Was the teeth originally attached to the jaws? Or not?
Basically,
Okay.
periodontal ligaments do flex a little bit, but on the order of this resolution, we can say this. But if we, so if we have dental implants already, fillings or something, yes. The other thing is that if we could put like wax on the side of the teeth and put a couple of markers there just for a little bit and then CT scan them right away and then take those off, in theory that could work. There's also been a bunch of research using tracking without markers. So I was part of a knee jump cut study in graduate school where they were using the shape of the tip of the tibia and the end of the femur to actually what they did was they took the CT scan and then they created virtual fake radiographs in different positions, and then they matched those with the video data. Is possible, yes, way more laborious. So on the time that I got out 5 papers, like maybe one of those papers came out. So it's a lot more work, but again, you've got a trade-off, because then you have this hugely relevant. We were looking at anterior cruciate ligament ACL reconstructions, and yeah, hugely possible. So it's not impossible in humans. It's just hard. Great question. We didn't really. That's a good question. We didn't really look at incisors much, mostly because we were studying food breakdown, like chewing, grinding up of that food. We do have data from rats of like when they have a big pellet and they're like taking bites of it. And so totally we could. And that's, again, one of the beauties of how XROM works is we can go back to that same data set next year or 5 years from now, when I have a student who asks that same question, and we can look at it again. Yeah.
I have just a question about these. You showed 6 models for human jaw stuff.
Mm-hmm. They've all been used in clinical settings for TMD research.
So, and you said, so I think I'm asking about, like you said, like people think that pigs are the best for TMD or something. Are you surprised by the animals we're looking at? I mean, are there other monkeys out there with faces more like ours? I'm just thinking like some little round-headed South American monkey that's got like big eyes. Yeah, so.
Yeah.
Yeah. Why are we looking at these animals?
Why are these ones? Good question, John. So I think that the best answer I can give is so much of this is a marriage of convenience. We have tons of colonies of mice that we can do transgenics on because they've been a model for everything about humans forever. So it's really easy and it's really cheap to get a whole colony. In this room, I could have a colony of 12,000 mice. No problem. Rats also used often in behavioral experiments, so we're used to them. Animal care is used to them. Rabbits, again, easy to come by. Macaques are a really common neuroscience monkey, so there's been a lot of work. And again, we've sort of worked out the animal care of dealing with them, and they're pretty darn close. Maybe there's some others that would be a little bit closer to humans, but they're pretty darn similar. But again, there's a trade-off. Super expensive. It's like $1,000 a day to keep a monkey in an animal care facility. It's like $1,000 a week to run a rat company. So, and also there's a lot of ethics in the whole monkey thing. I don't think I could do it all the time. And these are farm animals, right? We have lots of pigs, we got lots of sheep, like no worries. And so oftentimes it's the first pass is just what do we have? Starting a research system by saying I need to go get eyes from Madagascar, like, okay, have fun, bro. Like, it'll take a while. It's much easier to say I'm just gonna go spend $50 and get paid. The other thing I would say is that a lot of TMJ and TMD research happens in pigs because they're a really good size, and the morphology is relatively close. It's not perfect, but aside from that eminence difference, a lot of the force vectors for the muscles and the anatomy of the disc itself is very similar to humans. So it's mostly convenience. And I think part of it for me over the last few years of what's gotten me into this research is that I want to like ring some bells and be like, hey, all of the clinicians are treating these like they're all humans of a little bit different shape. And like, it's a lot more nuanced than that. And so I'm sort of on this crusade of teaching comparative anatomy to the clinical community, or trying, and I don't think a lot of years, but I try. I gotta do something.
Okay, there are no more questions. Another hand for Dr. Gidmark. Thanks everyone for coming. If you have time now, we're going upstairs and try to eat together and get to know Dr. Gidmark a little better.
Talk about our complex parts.
Yeah.
All right.